Hi everyone and welcome back to Neuropsychology. So we're going to go to the second video, and in the last video we talked about what is emotion, some basic components of emotion, we talked about some theories including the Papez theory, we talked about the Clive-Bussy syndrome, we talked about regret and some other limbic structures in the limbic system. So in this video, we are going to talk about the 6 universal expressions, cognitive emotional interactions, how we process an emotional stimulus in our brain, the social brain, frontal lesions in monkeys, cerebral lesions in humans related to emotion, social neural networks, and self-awareness. Okay, so people universally recognize some basic facial expressions. And this is based on research done by Paul Ekman and his colleagues. And they demonstrated that throughout the world, people universally recognize 6 basic facial expressions. So these are anger, fear, disgust, surprise, happiness, and sadness. So there are some cultural differences in the details of these facial expressions. And this suggests that the expressions of emotion can contain nonverbal accents that can identify the expressor's nationality or their culture. So overall, there are 6 basic facial expressions, but there can be tiny subtle differences between cultures, which are technically nonverbal accents. There was a researcher, Joseph Ledoux, and he chose to study one specific emotion. So before him, people were studying emotion as a whole, broad, holistic approach. But he chose to study one specific emotion, namely fear, as an exemplar of how to study brain behavior related to emotion. So in Ledoux's view, all animals inherently detect and respond to danger, and the related neural activity eventually evolve to produce a feeling, which in this case is fear. So you have danger, you create fear. So an example is when a mouse detects a cat. So in most situations, prey animals' fear is either related to having a predator nearby or to danger from others, which in this case is a cat. So he's afraid the cat's gonna catch him or eat him or play with him. So he sees a danger and then creates an emotion, in this case fear. For humans, our fear is a much broader emotion. So we face wide-ranging physical and physiological dangers, from sport injuries to, for example, things as big as terrorism. And we also have more subtle dangers, such as those posed by, such as chronic stress. So an important implication of Ledoux's theory is that our fear system includes both unconscious fear responses and our conscious awareness of feeling fear. So he presumes, however, that the neural system underlying fear is similar in both unconscious and conscious responses, and that the neural basis of fear can be studied using a model system, which in this case is fear conditioning. And we talked about that in a previous chapter. So a key brain structure that is developing a conditioned fear is our amygdala. And the amygdala sends outputs to stimulate hormone release and activate the autonomic nervous system, and therefore generates emotion that we can interpret, in this case, as fear. So psychological measures of fear conditioning can measure autonomic functioning, such as heart rate or respiration. And quantitative measures can measure behavior. So for example, standing motionless after the tone is heard, in the case of the fear conditioning model we talked about previously. So let's look at the image to the right. So information about an emotion valence stimulus travels from the sensory thalamus to the amygdala and the cortex. So then the cortex will feed back to the amygdala, where several projections initiate stress hormone release, activate the autonomic nervous system, or evoke emotion and suppress pain, and stimulate arousal and attention. So you see those all the way here at the bottom as well. I just put them in a nice little row right here. And then we have the hippocampus, which provides information related to context. So the hippocampus is really important to create this fear memory. And damage to the amygdala can interfere with fear conditioning, regardless of how it is measured. So people with temporal lobe damage that include the amygdala are impaired at fear conditioning. So imaging studies show that activation of the amygdala during fear conditioning, so whenever they learn to respond to a tone after fear conditioning, they have increased activation in their amygdala. So how does the amygdala know that a stimulus is dangerous? So in Ledoux's view that we talked about 2 slides back, there are 2 possibilities. So both implicate neural networks, one genetically involved and one is shaped by learning. So genetically based neural networks in the amygdala evolve with the animal. So rats born in the laboratory show fear responses whenever you play a sound of an owl, even though they've never encountered them. And they're born in the labs, so often you can take them away from their mother. They cannot, the mom can't tell them about it. So it is genetically inside of them. Most primates also show an intense fear of snakes on their first encounter, which suggests that they kind of have a snake detector that has evolved to sensitize us to stimuli associated with danger. So if you remember from a couple lectures back, we talked about John Downer's split brain monkey, and they had the amygdala removed. So the one that had the amygdala removed had no fear of a snake from the side of the brain lacking the amygdala, but showed an intense fear from the intact side, when the amygdala was still there. So this shows that the amygdala is very important in creating your fear response. So neural networks based in the amygdala likely also learn from experience about dangerous stimuli of which evolution could not prepare us. So we may have learned, for instance, that a person wearing a certain type of insignia, such as a characteristic of a violent gang, could be potentially dangerous. And a person wearing other insignia, such as a nurse uniform, is typically not dangerous. So Ledoux proposed that these circuits in the amygdala interact with cortical circuits to influence affective or our emotional behavior. So an important aspect of fear is obviously context. So a particular stimulus can be dangerous in one setting, but not in another. And this distinction is clearly important to our behavior. So for example, a highly poisonous snake is extremely dangerous when suddenly encountered on a pathway, but presents no danger to us when it's behind a glass wall at a zoo. So also, environmental context may acquire emotional properties through prior experience. So through classical conditioning, for example. So if a poisonous snake or if multiple snakes are repeatedly encountered on a path you take through the woods, then this path itself becomes threatening. So if you know, every time I go on this path, I see all these big snakes and they're poisonous, then at some point you're going to say, okay, this path is dangerous to take. Okay, so the interdisciplinary field of social neuroscience seeks to understand how the brain mediates our social interactions. So although advances in imaging technology tempt us to think that major insights into social brain no longer require lesion studies, it has been shown that lesion studies do not always support conclusions drawn from non-invasive imaging. So we technically need both. And we need both and try to combine both for analyses. So we need both lesion studies and non-invasive imaging to make correct conclusions. So next up, we will review some lesion studies in monkeys, and then some lesion studies in humans, and then we'll review research that talks about fMRI activation in healthy participants. Okay, so it is often reported that relatives report personality changes in patients that have brain damage. But the parameters of these dangers have been poorly understood and specified in humans. So, results of research on animals, particularly on non-human primates, however, they made it possible to identify 6 behavioral changes that are associated with emotional processes after frontal lesions. So, here you have the 6 things that happened. We're going to go by them one by one. So the first one is when these animals or monkeys had frontal lesions, they had reduced social interaction. So this was especially after orbital frontal or anterior cingulate lesions. And monkeys will become socially withdrawn and even fail to reestablish close relations with family members. So the animal will sit alone, and will not engage in social grooming or have contact with other monkeys. And in free-ranging natural environments, they will become kind of solitary, or they can even leave the troop altogether. The second thing is these monkeys will often have a loss of social dominance. So monkeys that obtained orbital frontal lesions and that were formerly dominant in a group, they then will fail to maintain this dominance. So although this decrease of dominance will sometimes take weeks, it kind of depends on the aggressiveness of the other monkeys in the group. So if other monkeys are trying to gain dominance quickly, this will happen faster. The third one is inappropriate social interaction. So monkeys with orbital frontal lesions will fail to show appropriate behavior to show that they are submissive to dominant other animals. So they also approach other animals without hesitation, irrespective of what the animal's social dominance is. So this behavior often results in aggression from other dominant animals with brains that don't have lesions. And similarly, when approached by dominant animals, monkeys with frontal lesions may simply ignore them or run away rather than performing typical submissive gestures. Okay, then we have number 4, which is altered social preference. So when a healthy monkey is released into a large enclosure that has members of the same species behind a glass barrier, normally it will kind of sit against the glass next to the animal sitting on the opposite side. So although these animals normally prefer to sit beside intact monkeys with no lesions of the opposite sex, monkeys with large frontal lesions prefer to sit with other frontal lesions monkeys of the same sex. And this is presumably because they are less threatening. And number 5 is reduced affect, so less emotion. Monkeys with frontal lesions largely abandon their facial expression or posturings and their gesturings in social situations. So these monkeys with frontal lesions will show a drastic drop in the frequency and variability of facial expressions and kind of always have like some form of poker face on. So definitely remember that. So the poker face is an example of reduced affect due to a frontal lesion in monkeys. So this is not a simple loss of facial muscle control because it can still produce these expressions. They just don't really do it. Okay, and the last one is reduced vocalization. So lesions of the frontal cortex reduce spontaneous social vocalizations. And this is often after anterior cingulate lesions. And if you take their entire anterior cingulate lesion, the entire cingulate cortex out, rhesus monkeys effectively make no typical vocalizations at all. So, these changes in emotional processes in monkeys with frontal lesions are especially interesting because they suggest that similar changes might be found in humans with frontal lobe injuries. So, in humans, frontal and temporal lesions result in deficits in producing facial expressions as well, but also in social speech, impair the perception of facial expressions and emotions relative to specific social context, and they can also change personality. So, this is specifically seen in when people cannot show the right facial expression in a social setting. So, for example, if you're at a funeral, you're supposed to have a certain facial expression which shows your emotion. You cannot laugh or smile. Rather, at a graduation, you're supposed to be happy and you're not supposed to be crying, right? Maybe crying of happiness. So lesions to the insula, which is the area right here in green, you can see it in the picture to the right. So this doesn't only increase the pain threshold, but it also impairs our ability to recognize pain in someone else. So we have a lack of empathy for pain. So this finding is consistent with imaging studies that showed increasing activity in the insula during tasks which were assessing empathy of pain. And amygdala lesions, all the way at the bottom, amygdala lesions impair the recognition of fear in others. And case studies also show that bilateral damage to the ventromedial, aka ventral, bottom, medial, towards the midline, that these regions in the prefrontal cortex produce impairments in social conduct, decision making, and emotion processing. So patients with bilateral ventromedial prefrontal lesions are also described as having poor judgment regarding their personal and occupational affairs and making poor decisions in laboratory tasks that are designed to measure complex decision making. Most case studies have also focused primarily on patients with bilateral lesions, largely because such patients usually have ruptured aneurysms of the anterior cerebral or anterior communicating areas, which lead to bilateral lesions in both hemispheres, or from surgical treatment of midline tumors, which also leads to bilateral injury. So finally, isolated case histories suggest that there are social changes related to lesions in the temporal parietal junction, or the TPJ, and the anterior cingulate cortex that we talked about a little earlier. And of course, we already talked about this, but lesions in the amygdala result in impaired fear conditioning, and we talked about that before. Okay, so let's talk about the social neural networks. So to identify brain regions implicated in social behavior, the scientists Kennedy and Adolphs, they reviewed lesion studies in brain-injured patients and fMRI activation in healthy participants. So there are 4 social-related brain networks that we're going to talk about next. And these are the amygdala network, the mentalizing network, and the empathy network. And then the last one is the mirror, stimulation, or action perception network. Okay, so let's start off with the amygdala network. So the amygdala network includes the orbital frontal cortex and the temporal cortex, as well as the amygdala. So this network's functions range from triggering emotional responses to detecting socially relevant stimuli. Next up, we have the mentalizing network, which is in blue, and this contains the superior temporal sulcus, as you can see right here, and the anterior temporal cortex. And the mentalizing network is a collection of structures related to thinking about internal states of others. So this network provides a mechanism for understanding the actions of others. And then the third network is the empathy network. This network involves structures that are recruited when individuals empathize with others. These structures include the insula and the cingulate cortex. You can see that here in blue. So here you see part of the insula. And here you see the cingulate cortex. And the empathy network can attribute to intentions of others, something we humans automatically do. And humans seem compelled to attribute intentions to other psychological motives, even to non-human and abstract animations. So we can either have empathy for, let's say, if one square was attacking another square in a cartoon. And the last network, the mirror stimulation action perception network, this is activated when observing actions of others. This network includes the mirror neuron system, so definitely remember that, the mirror neurons. We talked about that in 2 chapters already. And you can see that here in green. So you can see it on the lateral surface right here, and in the frontal cortex and the parietal cortex. So, it is all involved in developing our concept of self, which we're going to talk about in later slides as well. So here we've only talked about the cortical components of these social networks and a little bit of subcortical, such as amygdala, but other subcortical regions, such as the nucleus accumbens, the globus pallidus, the hypothalamus, and the VTA, or the ventral tegmental area, are most likely also involved. Okay, a little recap. Here is the amygdala network, has the orbital frontal cortex, amygdala, temporal cortex, and you can see that here in red. And here you have the amygdala. If you go back towards the brain, you can see the hippocampus right there. The mentalizing network you can see here in blue, so it's around the superior temporal sulcus right here, and also the front part of the temporal cortex. Then we have the empathy network, which is in blue. Here you see the insula, and here you see the cingulate cortex, and the action perception or mirror or stimulation network, which is in green. And you can see that here in the parietal and in the frontal cortex, are also called the premotor cortex right here. Okay, the last slide of this chapter. So we humans are aware not only of the actions and intentions of others, but also of our own actions and intentions. So we are self-aware. And there are 2 distinct neural networks in the frontal lobe structures that appear to be critical for generating our self. So one, we have a right frontal parietal network that kind of overlaps the mirror stimulation action perception network, and we have a cortical midline network. So humans and apes have a unique ability to recognize themselves, and this is called the self phase. And we can recognize ourselves in a mirror. So the right hemisphere of a split brain patient can recognize the self phase, and the physiological reaction to the self phase is greater for the right than the left hemisphere. So both imaging and patient data provide evidence that the right frontal parietal network controls recognition of the self phase. So recognizing yourself in a mirror. So researcher Lucina Udin, she does actually great neuroscience work right now at and her colleagues, they showed that the self-phase recognition activates right frontal and parietal regions, as you can see in the image to the right. And these activated regions overlap with regions that contain mirror neurons. So the activated neurons have been proposed to provide a link between self-perception and associated mental states of the self and understanding others' intentions. So Udin and her colleagues, they proposed that this right frontal parietal mirror neuron areas, that they act as bridges between self and the other. So if you see yourself versus others, so you can connect the two. They do this by co-opting a system for recognizing the actions of others to allow for recognition of the actions of yourself. You kind of compare. So self-recognition does not only involve faces. In another study with voices, Udin's group showed that listening to your own voice also increases activity in the right inferior frontal gyrus. So they conclude that the inferior right frontal region may contribute to a multimodal abstract self-representation. And action of the self are only part of what we would call self-awareness. Okay, and that was the end of this video, and I will see you guys back soon for the next chapter.